Hello students, welcome to EPG Pathshala. I am Professor Ramesh Kothari from Saurashtra University Rajkot, Gujarat. Today we are going to discuss about Prelude Carbohydrate Part A, in which we will study monosaccharides and oligosaccharides under paper Carbohydrate Metabolism. So students, let us see what we are going to learn in this module. Introduction and function of carbohydrate, to understand classification of carbohydrates, to study the characteristics of carbohydrates, and to understand the property of monosaccharides and disaccharides. Carbohydrates are the most abundant biomolecules on the earth. Carbohydrate is one of the four major class of biomolecules along with protein, nucleic acid and lipids. They make up most of the organic matter on earth because of their extensive role in all form of life. First, carbohydrate serve as energy source, energy store, fuels and metabolic intermediates. Second, ribose and deoxyribose sugars form part of structural framework of RNA and DNA. Third, polysaccharides are structural elements in the cell wall of bacteria and plants. In fact, cellulose, the main constituents of plant cell wall, is one of the most abundant organic compound in the biosphere. And fourth, the carbohydrates are linked to many protein and lipids where they play key role in mediating interactions among cell and interactions between cells and other elements in cellular environment. A key related property of carbohydrates in their role as a mediator of cellular interactions is the tremendous structural diversity possible within this class of molecules. Carbohydrates are built from monosaccharides, small molecules that typically contains from 3 to 9 carbon atoms and vary in size and in the stereochemical configuration at one or more carbon centers. These monosaccharides may be linked together to form a large variety of oligosaccharide structure. The unraveling of this oligosaccharide structures, the discovery of their placement at specific sites within proteins, and the determination of their functions are tremendous challenges in the field of proteomics. Functions of carbohydrates. Carbohydrates function as biofuel, primary source of energy, storage food, framework in body, anticoagulants, antigen, hormone, provide raw material for industry, act essential element for fat oxidation, play an important role in gastrointestinal function, add flavor to the several type of diets, and play an important role in molecular recognition. Classification of carbohydrate. The word saccharide comes from Greek word saccharon, meaning sugar. Those carbohydrates such as glucose and fructose that cannot be hydrolyzed into simpler compound are called simple sugars or monosaccharides. Certain carbohydrates are made of two molecules of monosaccharides such as sucrose, lactose, and maltose. These carbohydrates are called disaccharides because maltose is made up of glucose and glucose, lactose is made up of glucose and galactose, and sucrose is made up of glucose and fructose. Similarly, trisaccharides are composed of three and tetrasaccharides are composed of four monosaccharide molecules. All these compound carbohydrates may be readily hydrolyzed into their constituent monosaccharide by heating with dilute acid so or by action of specific enzymes. According to the number of carbon atom and functional group in its simplest form of carbohydrates are further classified. The suffix OAC indicate the type of sugar. The classification of carbohydrates based on the carbon atom it contains, if it contains three carbon atoms, it is known as triose, tetrose, and pentose, hexose. Likewise, as the number of carbon increase, the nomenclature also change. Nomenclature of carbohydrates based on number of carbon atoms. Triose, three carbon atoms. The examples are glyceraldehyde and dihydroxyacetone. Tetrose, if carbohydrate consists four carbon atoms. Here example is erythrose. Pentose, a carbohydrate that consists five carbon atoms, and examples are ribose, ribulose, and xylulose. Hexose, a carbohydrate which consists six carbon atoms. An example are glucose, galactose, mannose, and fructose. Heptose, a carbohydrate that consists seven carbon atoms. An examples is pseudoheptulose, and so on. As the number of carbon atom increase, the nomenclature also change. So if you see the nomenclature of carbohydrate, carbohydrate can be categorized under four categories: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. In monosaccharide, there is one sugar molecule. In disaccharide, there are two sugar molecules. In oligosaccharide, there are 2 to 10 sugar molecules. And in polysaccharide, there are 10 or more sugar molecules. If you see the example of monosaccharide, as we discussed previous, it is glucose, fructose, and galactose. These are monomers. In disaccharide, you can say sucrose, lactose, and maltose. In all these three, glucose is the common monomer, and in sucrose, addition to glucose, there is fructose. In lactose, in addition to glucose, there is galactose, and in maltose, two molecules of glucose. In oligosaccharide, the example are raffinose and staclose, and in polysaccharide, the example are starch, glycogen, and cellulose. The simplest of the carbohydrates, the monosaccharides, are either aldehydes or ketones with two or more hydroxyl groups. Many of the carbon atoms to which hydroxyl groups are attached are chiral centers, which gives rise to the many sugar stereoisomers found in nature. The two family of monosaccharides are aldoses and ketoses. If the carbonyl group is at the end of the carbon chain, that is in an aldehyde group, the monosaccharide is an aldose, and if the carbonyl group is at any other position, that is in a ketone group, the monosaccharide is called a ketose. All the monosaccharide except dihydroxyacetone contains one or more asymmetric carbon atoms and thus occurs in optically active isomeric form. The common monosaccharides have cyclic structure. Monosaccharides are reducing agents. Disaccharides such as maltose, lactose, sucrose. Maltose, which contains two monosaccharides, that is glucose and glucose. Lactose contains glucose and galactose, and sucrose contains glucose and fructose. These two monosaccharides join covalently to form a glycosidic bond, which is formed when a hydroxyl group of one sugar react with the anomeric carbon of other. Characteristic of carbohydrates. The first characteristic, that is isomerism. Isomers are defined as molecule with the same chemical formula and often with the same kind of chemical bonds between atoms, but in which the atoms are arranged differently. That is, they have different structural formulae. Isomerism can be of two types: structural isomerism and stereoisomerism. In stereoisomerism, there are two types of sub-stereoisomerism. These are geometric isomerism and optical isomerism. Now, monosaccharides. A Greek word mono means one and saccharon means sugar. Monosaccharides are the simplest sugar formed by the single molecule, which further cannot be hydrolyzed into smaller units. Depending upon the number of carbon arrangement in a unit, monosaccharides are subdivided into aldoses and ketoses. The aldoses and ketoses are represented by the following general formulas. Study of the chemical and physical properties of many sugar has shown the cyclic form predominate over open chain structure, both in solution and in the solid state. Alcohol may react with aldehyde and ketone moieties of the carbohydrate to form an unstable compound called hemiacetals or hemiketals respectively. This is a reaction between two molecules, that is, it is the intermolecular. If you see, when alcohol react with aldehyde, it produces hemiacetal and this reaction is reversible. Likewise, alcohol when react with ketone, it form hemiketal, and same way the reaction is also reversible. In glucose, the hydroxyl group on carbon 5 can react intramolecularly with carbonyl group on carbon 1 to form a stable cyclic hemiacetal. The cyclic form of glucose is a six-membered ring. Such sugars are called pyranos because they resemble pyran. Fructose form a five-membered ring called furanose as it is resemble furan. The pyranose and furanose ring structure of sugar proposed by Haworth more accurately represent their actual configuration than do the older projection structures used by Fischer and other workers. Through the older ring form are still used extensively in writing reaction of sugar. The pyranose ring of a 6-carbon aldose sugar can exist in either a boat or a chair configuration. The substitutes attached to the ring carbon that extend parallel to the symmetry are said to be axial, whereas those that extend outward from the axis are said to be equatorial. The axial is symbolized as A and equatorial is symbolized as E. Stereoisomerism. Many of the carbohydrates contains the same number of atoms and the same kind of group, yet definitely distinct substances. For example, the formula of C6H12O6 represent 16 differently simple sugar, all processing the structure CH2OH, CHOH, 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 and CHO. This is due to differently arrangement of the constituent groups of the molecule in space. This phenomenon represents what is called stereoisomerism, that is, space isomerism, and these sugars are stereoisomers. An asymmetric carbon atom is carbon when it is attached to four different types of atom or groups. When there are several asymmetric carbon atoms in a chain molecule and the end group are not identical, the number of stereoisomers possible is equal to 2:n, where n is the number of asymmetric carbon atoms. For example, glucose contains 4 asymmetric carbon and thus are 2 is to 4 equal to 16 isomer. The simplest monosaccharide is glyceraldehydes. Glyceraldehyde has a single asymmetric carbon atom, the central one, and so two stereoisomers, also called optical isomers as seen in the figure, are possible. That is, two forms of glyceraldehyde denoted as D and L glyceraldehyde as shown in the figure, which are mirror images of each other. It has been possible to relate the special configuration of sugar, amino acids, and many other optically active substances of biological importance to the configuration of dextro and levo form of glyceraldehyde. Monosaccharides can be named by a system that is based on number of carbon with suffix OSE added. There is also a system for numbering the carbon. They are numbered sequentially with the aldehyde or ketone group being on the carbon with the lowest possible number. Optical isomerism. When a beam of plain polarized light passes through the solution of a non-racemic mixture, rotation of polarization occurs. Carbohydrates contains asymmetric carbon. Because of this, carbohydrates are optically active and rotating the plane of polarized light. The compounds which rotate the plane polarized light to the right, that is clockwise, is called dextrorotatory, which is indicated by D or plus sign, and the compound which rotates the plane polarized light to the left, that is counterclockwise, is called levorotatory and indicated by either L or minus sign. Figure shows the presentation of change of path of light in when a polarized light pass through a solution. Enantiomers. An enantiomers is one of the two stereoisomers that are mirror images of each other and are non-superimposable. Epimers. Epimers are those sugar which differ with its isomer in configuration at only one asymmetric carbon. D-glucose and D-galactose, as shown in figure, contains four asymmetric groups, and that all these groups are identical except the configuration at carbon 4. Such isomers are called epimers. The process of interconversion of epimer is known as epimerization, and epimerases catalyze this reaction. There are several properties of monosaccharide. These are mutar rotation, glycoside formation, reducing power, reduction, oxidation with mild and strong oxidizing agents, methylation and esterification, dehydration, ozone formation with phenyl hydrogen. Glucose. Glucose is an aldohexose. It is a polyhydroxyaldehyde and contains 6 carbon. Carbohydrates are transformed into simplest glucose, which is the primary carbohydrate utilized by the body tissue. Glucose is the sugar or blood and other body fluids. Disaccharides like sucrose, maltose, and lactose, and polysaccharides such as starch, glucose, and glycogen contains glucose as one of the monomers. Glucose is 25% less sweet than sugar. Blood normally contains 60 to 90 milligram of glucose in each 100 ml. Galactose. Galactose is an aldohexose. It is a polyhydroxyaldehyde and a 6 carbon containing compound. It is found in dairy products, sugar beets, pectin, and gum. Galactose is one of the monomer of disaccharide lactose. Galactose is 80% less sweet than sucrose. It is epimer of glucose. Fructose. Fructose is a ketohexose. It is a polyhydroxy ketone and contains 6 carbon. It is found in fruit, flour, root vegetables, and honey. Disaccharide sucrose contains fructose as one monomer. It is the sweetest of all naturally occurring carbohydrate, 175% sweetened than sucrose. Fructose has higher solubility than other sugar. Properties of oligosaccharide. Oligosaccharides are composed of disaccharide, trisaccharides, and tetrasaccharides, and are so designated to indicate the number of monosaccharide units involved in the structure. Disaccharide is a sugar formed when a bond between the first carbon of one sugar and a hydroxyl group at any position on the other sugar is formed and a molecule of water is removed. Sucrose, maltose, and lactose are the most important disaccharides. Each one of these on hydrolysis with either an acid or an enzyme gives two molecules of monosaccharides by which it is made of. Sucrose. Sucrose, it is also called a stable sugar. Sugar occurs especially in the juice of plants such as sugar beet, sugar cane, sorghum, sugar maple, and pineapple. Ripe fruit are rich in sucrose. 20% of sugar cane is sucrose. It is non-reducing sugar, is relatively stable towards the action of alkali, and in general does not give the reaction characteristic of the sugar group, and hence no reaction with the Benedict's reagent. Hydrolysis of sucrose in an acidic solution or by enzyme invertase or sucrase results in the monomer, that is a molecule of fructose and glucose. Sucrose has alpha 1 2 glycosidic linkase. Lactose. Lactose is formed by mammary glands and occurs in extent of about 5% in milk, hence it is also called milk sugar. Lactose is hydrolyzed by acid and the specific enzyme lactase into its constituent monosaccharide glucose and galactose. It is reducing sugar, forms oxazones, a synhedrin and oxime, and it is decomposed by alkali. Lactose intolerance is a fairly common condition, means deficiency of lactase enzyme which break the glycosidic bond. Lactose has beta 1 4 glycosidic linkase. A water molecule and lactose is formed with the dehydration. Human milk differ from cow's milk in containing, in addition to regular lactose, other oligosaccharides such as L-fucosyl lactose, in which the deoxysugar L-fucose is linked to galactose at carbon 2 of the monomeric unit of alpha D-glucose and 1 beta galactose. Lactose is formed by mammary gland and occurs to the extent of about 5% in milk, and hence it is called a milk sugar. Lactose is hydrolyzed by acid and the specific enzyme lactase into its constituent monosaccharide glucose and galactose. It is reducing sugar, forms oxazones, synhedrins and an oxime, and is decomposed by alkali. Maltose. It is also known as malt sugar. Maltose is made up of two glucose units and is formed when the enzyme amylase or diastase hydrolyzes starch. Maltose is formed as an intermediate product in the acid hydrolysis of starch and consequently is an important constituent of corn syrup, which are prepared by partial hydrolysis of starch with dilute acid. Maltose is readily fermented by yeast. Maltose has alpha 1,4 linkase. Figure shows the alpha 1,4 linkage is present in the maltose. Cellobiose. Cellobiose is obtained as a product of the incomplete hydrolysis of cellulose. It is composed of two molecules of glucose into which it is split by either acid or the enzyme emulsion. It is reducing sugar and give the reaction characteristic of the sugar group. Cellobiose bears a same relation to cellulose as maltose does to starch and glycogen. Figure shows the structure of cellobiose. So, students, let us summarize what we have learned in this module. Sugars, which is also called saccharides, are compounds containing an aldehyde or ketone group and two or more hydroxyl groups. Monosaccharides generally contain several chiral carbons and therefore existing in a variety of stereo chemical forms. Monosaccharides commonly form internal hemiacetals or hemiketals, in which the aldehydes or ketone groups join with a hydroxyl group of the same molecule, creating a cyclic structure. This can be represented as Haworth presentative formula. Oligosaccharides are short polymers of several monosaccharides joined by glycosidic bonds, and at one end of the chain, the reducing end, is a monosaccharide unit whose anomeric carbon is not involved in a glycosidic bond.